Viruses are among the most protean entities in nature, ceaselessly mutating and acquiring new characteristics. These tiny entities follow a simple and relentless imperative: infect as many host organisms as possible. Occasionally, a virus' genomic alterations enable it to leap from one species to another, in a process known as spillover. In new research appearing in the journal mBio, Masmudur Rahman and his Arizona State University colleagues join international researchers to investigate one such spillover event, when the myxoma virus, MYXV, made a species leap from European rabbits to Iberian hares. Researchers would like to better understand these genomic transitions, as spillover events have profound implications for both human and animal health. Understanding the subtle alterations enabling viruses to make species jumps may help better prepare for outbreaks of new diseases, limit their transmission, and perhaps allow researchers to outwit viral mechanisms that set the stage for spillover events. In addition to its importance for the study of host pathogen coevolution, myxoma virus has been investigated for its remarkable ability to target and kill human cancer cells, while leaving their normal healthy cell counterparts unharmed. The new study suggests that the M159 protein not only enables MYXV-Tol to leap over the species barrier and infect hares but also appears to help this strain replicate even better in human cancer cells, potentially improving MYXV as a cancer-fighting agent. In the future, identifying the proteins that interact with M159 in hares and human cancer cells will allow us to understand whether M159 targets similar or diverse signaling pathways, said Rahman. Rahman is a researcher in the Biodesign Center for Immunotherapy, Vaccines and Virotherapy at ASU. He is joined by Grant McFadden, director of the center, and by Arvind Varsani, a researcher in the Biodesign Center for Fundamental and Applied Microbiomics. Collaborators on the new study include researchers from Universidade do Porto, Verão, Portugal, Universidad de Oviedo, Campus El Cristo, Oviedo, Spain, and IRIAF, CIAG del Chaparrillo, Ciudad Real, Spain. This fact is due to a historical event in which MYXV was used to control populations of European rabbits in Europe and Australia, beginning in 1950. Many kinds of viruses have spillover potential. Annual outbreaks of influenza, for example, are the result of spillover events occurring when migratory birds, acting as reservoirs for the virus, spread the disease to other species, including ducks, chickens, pigs and humans. As the virus moves from species to species, mutating strains acquire new abilities to aid their transmission and ability to evade host immune defenses. Although the natural hosts of the MYXV virus are Silvilagus rabbits, known in the Americas as cottontails, exposure of European rabbit populations to this virus proved 99% fatal, without any further adaptation of the virus to the European rabbit host. The highly contagious virus, spread through rabbit populations by fleas or mosquitoes, produces a lethal rabbit disease known as myxomatosis. MYXV-Tol was found to cause a very similar lethal disease in hares. Over the long term, the rabbit control strategy with MYXV failed, as evolutionary selective pressures acting on both the virus and host resulted in MYXV-resistant rabbits and attenuated virus variants. In the case of MYXV-Tol, we learned that the acquisition of a single new virus gene allowed this new virus strain access to a new host species that was previously resistant to the virus. New virus on the block evidence suggests that Iberian hares had long been exposed to MYXV or a similar virus S. The new strain had acquired a small suite of new genes, which it acquired through recombination with an as yet unidentified poxvirus. The result was a supercharged variant that proved both infectious and highly lethal to hares living on the Iberian Peninsula, killing hundreds of them beginning in the autumn of 2018. The researchers examined laboratory cell lines of rabbit, hare and human cells exposed to MYXV variants with and without the M159 protein. While strains containing the novel protein did not appear more infectious to cells of European rabbits, the M159 containing strains were now highly infectious to cells from European hares, whereas strains without the protein were not, establishing M159 as the key ingredient allowing MYXV to cross Th. The study also tested two human cancer cell lines that are normally resistant to MYXV, exposing them to the M159 enhanced version. The results were dramatic. Human pancreatic cancer and melanoma cells are typically semi-permissive or non-permissive to MYXV, meaning that the virus usually replicates poorly in these cell types. Further research promises to shed new light on the highly pathogenic MYXV-Tol variant as well as illuminate the mechanisms used by other pox viruses to spill over into new animal species, including humans. Identification of a novel myxoma virus C7-like host range factor that enabled a species leap from rabbits to hares. mBio. 2022. 13, 2, doi. 10. 1128, mBio. 03461, 21 Arizona State University. Crossing barriers. How the rabbit virus myxoma leapt into a new species. Science Daily, the 26th of April 2022. ift.tt/ethnolv. Arizona State University. 2022, April 26. Crossing barriers. How the rabbit virus myxoma leapt into a new species. Science Daily. Retrieved April 26, 2022 from ift.tt/kg86bs Arizona State University. Crossing barriers. How the rabbit virus myxoma leapt into a new species. Science Daily. ift.tt/imulkihai. Accessed April 26, 2022. Thanks for watching. Please subscribe my channel for more videos.